wall of nature and nurture. How genes and environment combine to affect our life course. Dalton Conley, New York University.
When the wall came down in the fall of 1989, I didn't hear anything about it because my grandfather had just been struck down in an accident in Miami Beach, and I was spending day and night supporting my grandmother.
Good morning. Thank you, Sebastian and everybody else for coming to hear me speak. I have a little pet theory. This is not my own research, just my hobby, that all academics are studying themselves in some way. So I imagine that Tanya started looking at emotional compassion because she's an identical twin. I started looking at nature and nurture, I think, because of my own childhood, which I want to start with. Childhood natural experiment, I call it. Sorry about these slides. This on the left, my left, your right. I'm sorry, your left is the neighborhood where I grew up in, and it was a low-income neighborhood, what we call a ghetto of Puerto Rican and African American families, except for my sister and I, who you can see in the picture. You can guess which one is me, I think. As we grew up there, the children of artists, we ended up, not understandably perhaps, with very different outcomes from our neighbors. So here I am, standing before you, having the great honor of giving this lecture, and that's me playing the clarinet very badly in 7th grade. And there was my best friend from the yearbook, who shortly after that photo was taken, was shot in the neck and paralyzed from waist down for the rest of his life. And I think that's what's motivated me in my own research to ask, why did we have these different outcomes? And little did I know, I was getting into one of the great debates in the last 150 years, which is nature versus nurture. So on the one hand, we have the mouse that represents the behavioralists who think that any behavior or any personality, any aspect of our lives can be socialized, can be taught, famously, as argued by Watson. And then we have the other Watson and Crick, who discovered DNA and argued that led to an argument that most of what we do, say, believe, our personality, is innate, is part of the genes. And I want to argue today show you some evidence that that's a false dichotomy. Now, sociologists are fond of saying, yes, how genes matter depend on your environment. If you have a gene for aggression, let's say, and you're born in the ghetto like me, you end up in jail. But if you have that same gene for aggression and you're born into a very wealthy family, you end up as CEO or executive. But that's how do we get from that simple proverb to a real empirical strategy to understand gene-environment interactions? And in fact, I want to make a broader claim that actually the genes of those around you are part of your actual social environment. But let me tell you a little story of how I got here. So as a sociologist, we can't really run experiments to answer our questions very well, because I can't randomly assign half of this room to become married and the other half to get divorced and then follow you. So my ideal experiment would be to take two twins like this, maybe you'll volunteer, and take them at birth or even before birth and separate them and raise them in randomly assigned households. And then I would have to be a stalker, hide in the bushes for about 35 years or 40 years, watching them and taking detailed notes to know how they turn out. And then I'll know the effects of nurture as separate from nature. Of course, I can't do that, but there is some new promise with the rise of molecular genetics, the sequencing of the genome, and the identification of polymorphisms, markers that are distinct within the human population that vary from person to person. And I want to talk about one in particular today, but I have to explain a little of what I mean. So there are two types of main types of polymorphisms of variation. One is what's called a SNP, a substitution at a single point of one base for another, in this case T to A. And there's another kind, there are multiple kinds, but the two main kinds are, the second is a microsatellite or variable number tandem repeat. And that's where, imagine if you had to copy the same sentence over and over and over again, you'd probably make a mistake of how many times you wrote it. So it's a very common error in replication to have repeats like this of different numbers in different people. And that's the kind I want to talk about today. In particular, I'm going to talk about the serotonin transporter. How many of you are on Prozac or some other? No, you don't have to answer that. Serotonin transporter is that protein on the right that allows the serotonin to be reuptaked back into the presynaptic neuron and therefore be used again. And the SSRIs, or the Prozac, slows that process down and leaves more serotonin in the synapse. There are two major alleles in the human population of the promoter region, which is not the shape of the protein itself, but the regulatory region. How much of this protein are you going to produce? How many of these transporters are you going to have? And there's the long allele and the short allele. And the long allele, think of it as like a helicopter landing pad. It's a bigger promoter, therefore it's easier for it to be transcribed, and you get more of the transporters, and the short allele gives you fewer. And it's been observed in animal studies that those with the shorter allele, or this gene knocked out, end up with a phenotype of social defeat, depression, anxiety. Yes, they can measure all this in mice, and they find these very depressed mice. So it was when it was discovered in animal models that this had this important outcome, people started looking at it in humans. And at first, it was thought the short allele was bad, right? It led to this depression, it led to this negative phenotype. But then that left us with a mystery story. And it's a mystery that takes place over 40 million years or so, because we share this variation of short and long with our relatives, as you can see, all the simians and prosimians. And in fact, what makes the mystery even more interesting is the fact that the promoter has gotten shorter and shorter over evolutionary history. In other words, the bad allele has been getting more frequent as we've, quote, evolved. And so humans have the highest rate of the short, quote, bad allele. So that led some people to say, well, maybe it's not bad. Maybe we're conceptualizing the wrong way. And they started this debate, or this concept of the orchid versus the dandelion. So an orchid is a beautiful flower, but you need to take very good care of it, like a petite prince, in a jar, perfect temperature, everything. And if you do that, if you give it the perfect environment, then you get this beautiful flower. But a dandelion, so I like to think of it as we're in Germany, make fun of the French. In humans, the idea is that it's an emotionally reactive, emotionally sensitive allele. So that's why maybe it stuck around. It gives you sensitivity to other people's moods and emotions. It makes you reactive. But if you're a dandelion, as No Drama Obama probably is, that you're cool and calm and you do not react. But you don't look as beautiful. I don't know if Barack is more beautiful than Nicholas or not, but you can survive like a dandelion and you look the same, and it's across all environments. Like dandelions can live in many environments. So that concept got very popular in the psychological literature to explain, to make it more complicated, the story of the fact that there is these two alleles, and it's not just negative to have the short one. And this parallels literature in evolution in biology, which is called phenotypic capacitance. So is this serotonin transporter a phenotypic capacitor? And this comes from writings 50 years ago by Waddington that argued that there are certain genes that mask variation, And then when they're released, or when they're compromised, you get a radiation of different phenotypes as an evolutionary strategy to survive. So in other words, you uncork the genie bottle, and all these different creatures come out, and at least in a challenging environment, hopefully one of them will survive. So I looked at twins. I don't think Tanya was in my study, but twins we could separate out into those who share their genome completely and only have differences in their environment, and twins who have both differences in environment but also differences in their genome, i.e. fraternal twins that are the same sex. But when we do that, we find that indeed those with the short allele end up more different in their depression. Not the average level, but the difference between the two siblings is greater when you have the orchid allele than when you have the dandelion allele. And when we separate that out into two genetically identical monozygotic twins and dizygotic twins, we see the same pattern. So that explains that maybe there is something to this allele orchid dandelion hypothesis. However, that doesn't really answer the mystery of why has this remained in balance. Why don't we all have the short allele? Why is it that at some level of group selection do we need both in the population? And there you turn to another theory of evolution, which is called frequency-dependent selection, or balancing selection. And we can think of many examples of this in nature. One is apostatic selection. I show you these multicolored fish because evolution actually preserves the range of variety in their morphology and their color because it increases the search costs for their predators. If the fish are all the same color, it's easier for a predator fish to hone in on them, but when they have different colors, it's more confusing. Likewise, on the right is an image of the major histocompatibility complex protein, and we have a lot of variation in that, and the idea is that at least some of us, if there's a major plague, at least some of us will have the right one to survive and repopulate the Earth. So maybe this is happening on a behavioral level as well, I asked. Maybe there is balancing selection going on for these particular alleles. And you could think of a game theory, like the games that were described by Tanya, that maybe it's actually good to be the grumpy, demanding Sarkozy baby if you are going to get a lot of attention and your siblings don't have that allele. They're calm, you're screaming all the time, and you get more investment from your parents that way. Or maybe it works the other way. Maybe when you're the bad kid, or the demanding, screaming, loud, sensitive kid, then your parents shun you, and you end up worse off. But the key to this theory is that your genotype alone does not matter, that your genotype only matters in the context of your family's genotype, and particularly your siblings that you're in competition in the nest, if you will, with. So again, I looked at twins, and I looked at, in this case, not identical twins, but only fraternal twins. What did I find? I found that indeed your allele, if you have 0 of the short alleles, it doesn't really matter what your siblings are in terms of their alleles. But if you have, if you have the long alleles, and you have no short alleles, if you have the short, sensitive, reactive alleles, then it does matter what your siblings do. And in fact, if you're all a bunch of whiny, difficult babies, then you're fine. You're all whiny. But if you're the only one, then you get less parental investment, and you end up worse off in your mental health and your psychology as you age. So my final sentence is that our environment is not just the uterus we were gestated in, it's not just the toys and books and our colleagues around us, it's the very genes around us, and therefore there is no wall between nature and nurture. Thank you.